It is a real honor and privilege to be able to speak to you today. And as Colin was saying, it's kind of amazing. I started today in Barcelona, and I'm going to end today actually in San Francisco. It's an amazing world that we can actually do that. It's kind of like we're talking about big data, but we're in a small world now, actually, that the flights are just there to do this. And so I'm here to talk about big data and just what we've been doing, and I thank you all for making it all the way to the end of the meeting. And when I talk about big data, I'm going to talk a lot about omics technologies. I know you guys have had about 2 days of lectures on omics technologies, but this to me is the most exciting reason to be in this field right now. We have these amazing tools. There's one on the slide here, gene chip from a company called Affymetrix. These tools that just quantitate so many different molecules, whether it's the RNA or DNA or proteins. This is just one example of these kinds of tools. And these tools have been with us now for more than 10 years. Scientists can use that chip on the left. It's deliberately made to look like a chip and quantitate, for example, all the different RNA blueprints in a cell or a tissue, disease, cancer, normal. And it's kind of funny, when we get tired of measuring them one by one, we kind of measure them on the right in 96 well format. So the chips that you see on the right, each well is one of these kinds of chips. And it's kind of funny how, in IT, we scale by 1024, right? Kilobyte, megabyte, gigabyte. In life science, we scale by the number 96. So if you're tired of 96 well plates, you go to 384 well plates, and then you go to 1536 well plates. So we use the number 96 to scale. Now, these chips are incredible. And again, it's just one example of this kind of omics technology. But can we believe we've had this for more than 10 years now? We've had scientists using these for more than 10 years. The price of these chips are a couple $100. Quantitate every gene in the genome, put a sample on there. And so whereas everyone started to use these chips, people, scientists, whether they're in Boston or Stanford or anywhere else, start to write papers about their findings. Here are genes, there are genes. This cancer seems to have more of these genes. This particular form of diabetes has fewer of those genes. And they started writing papers and submitting papers about it. And then it was the journals that started first. And then it was the funding agencies that said, we are getting too many of these kinds of publications. We need to start releasing this data to the public. And so where are we today? As of August 1st last year, we have 1 million of those chips publicly available. As nifty and amazing as those chips seem, the public can now go and access 1 million of those samples. And you see the growth chart on the right here on this slide, the bottom right. And you can see we started essentially from 0 in 2000 to now public repositories with 1 million samples. And it's kind of sad. It looks like it's slowing down there at the end, if you see all the way at the right there. And then you realize, actually, they only counted half the year because this came out in August. We're still doubling about every 3 years or so the amount of data. It's just an unbelievable trend. And so just to make it painfully clear, because people ask me about URLs all the time, where do I find these million samples? I'm just showing you 2 of the big screenshots here. One is from the National Center for Biotechnology Information. It's in the National Library of Medicine. It's one of the institutes of National Institutes of Health, NIH. And even there, if you go to the Gene Expression Omnibus, you can find 929,000 samples there. The Europeans run one in the background there, another 250,000. Actually, if you look today, we're already at 1.25 million samples. 1 million was last year. That was eons ago, right? We've already added another 250,000 samples here. And just to make this painfully clear, and I know many of you have maybe seen me say this on YouTube videos, just to make this really clear, a high school kid today, even a high school kid today that needs to do a science fair project, she can go to this website here and type in the word breast cancer. Just search for breast cancer because she's got to do a science fair project. And now today, she can go and download 39,000 samples of breast cancer about as easily as she can find a song on iTunes today. If you go and click on this website, you can go download the digital form of 39,000 samples of breast cancer and do whatever science you want on that. That's not future tense, that's here, that's today. And if for some reason that's not enough for you, in 2 years, it'll be double this number again, right? If you're still not happy with that number. Now, 39,000 samples sounds kind of interesting. Actually, the number I really like is the top number there, 1,500 experiments. 1,500 experiments here. What does that mean, 1,500? So, 1,500 labs, 1,500 groups, 1,500 experimenters generated data and deposited it into this repository. Let me tell you how crazy that is. Imagine I'm starting a brand new effort to study breast cancer today, okay? And you're the funding agency. And I go to you and I say, you know what? I'm not going to study breast cancer myself. I'm going to get my friends on the internet to help me. And the kids today call that crowdsourcing, right? So I'm going to crowdsource breast cancer research. And I'm going to get all my 1,500 best researchers in breast cancer to run the best samples they have, the best patients they can find, the most latest technologies in each of these labs, and they're going to generate all this data. I'm going to have them all give me all of their data for free, because I'm not even going to pay them. Now, if you were the funding agency for this, you would laugh me out of the room. Why would these 1,500 labs give you any data at all for no money, right? Yet here we are today. 1,500 labs, the best in the world, have seen their patients, have generated their samples, and put it out on the internet. So what I call this public open data, what I call it is actually retroactive crowdsourcing, okay? It's retroactive crowdsourcing. The crowd is helping you, and they don't know it. And the crowd is helping you, and you don't know it. But the samples are there, and they're just continuing to grow, waiting for you to do something with this. Now, if you have doubts that a high school kid can actually do this, I have to show this kid, okay? I've never met her. I hope to meet her someday. This is Brittany Wenger of Sarasota, Florida, who last year, at 17 years of age, had a neural network to diagnose breast cancer. She was the #1 winner in the Google Science Fair competition. This was about 2 years ago. So she indeed got public data, figured out a neural network, and that was the top prize in the country, actually in the world, for the Google Science Fair competition. And then last year, she actually figured out, that's breast cancer. Let's work on leukemia. And she did the same thing again. Leukemia was on the front page of many newspapers around the world. Yes, indeed, a high school kid can do miraculous things if we open up this data and let everyone get to it. Not just the scientists that generated it, but anyone get to this. Even a high school kid can do it. And of course, I'm saying, if a high school kid can do this, there's no reason any of you can't do this either. So in my lab, we start to just go like crazy with this. If so many people are running diseases and generating these arrays, my first big grant at Stanford is just go get every human disease. Go get them all. Let's just go get every disease that's been studied with these technologies and just curate them and make a master list, a grid here. And I'm showing you some papers, and I'm not going to even talk about the technologies involved, the computational text mining and normalization. But in the end, we kind of get a grid like this. So the way to read this grid is the rows going up and down are the diseases, like cancer or leprosy or HIV, and the columns across are all the different genes, 20,000 genes in the genome. If a gene goes up in a disease, it goes to yellow. If it goes down, it becomes blue. And we end up with a grid of all of these kinds of diseases here. And while we're collecting the diseases, so on the left here, you're seeing patients at the top in red. We get patients that have a disease. We have patients that don't have a disease, or individuals that are disease-free, and we start to compute a difference. What genes go up in this disease? What genes go down in this disease? We get what we call a signature. At the same time we were collecting this data, it turns out researchers, especially around here in Boston, were doing the same kind of experimentation with drug data. So they're taking cells, and they put cells with a drug, without a drug, with a drug, without a drug. So on the left, I have with a disease, without a disease. And on the right, I have with a drug, without a drug. And what we realize is all of this data is open access, public access. Why don't we marry these two together? And I'm going to make the statistics really simple with my arms in front of you here. If I've got a disease now where this gene goes up and this gene goes down, and I got a drug that can make this gene go down and this gene go up, maybe this drug and this disease should meet each other, right? And so the reporters call this Match.com for drugs, and the famous saying is opposites attract, right? So the drug has to really, of course we're not naive about these things. A drug has to get to the right part of the body. The tissue has to reach the tissue. Some drugs don't reach the brain very easily. There's got to be, if we want to commercialize these, there has to be intellectual property, there has to be formulation, there's got to be few toxicity from the drug. So there's a lot of thinking that has to happen, but in general, that's how the ideas start. And the first one of these we tried in the lab was actually this one, a seizure drug, an epilepsy drug called Topiramate. We predicted works on Crohn's disease, so a brain drug working on Crohn's disease. And you can see here my famous rat colonoscopy videos. And on the left, that's what a normal rat colon looks like, and probably your colon too. In the middle is a rat with Crohn's disease, inflammatory bowel disease. You can see the redness when it comes on the video. And on the right, you see what happens if you get the same rat with the Crohn's disease, the epilepsy drug, and seems to be all better. And so this was more than enough for us to pursue, and we've done many of these now, many, including this one, which I'm most excited about, which actually just came out today. So if you go to Fox News right now, you're going to see this in the story. I mean, this is literally coming out live. Why am I excited about this one? This is a disease called small cell lung cancer. It's got a 5% survival in 5 years. This is not a great disease to have. And on the left here, you see a mouse that has small cell lung cancer. You see the kind of nodules here and there. And on the right is the same kind of mouse. This is worked on by Julian Sargent, Alejandro Sucredero, my great friends and collaborators, where they treated the same mouse, the same kind of mouse, with imipramine, which is an antidepressant, okay? An antidepressant. It's not a antidepressant we use much anymore. We've moved on to newer ones, serotonin reuptake inhibitors. This one had some minor side effects. Nothing as bad as having lung cancer, though. And if you can see here, the lung cancer is melted away. It's gone. And why am I excited about this one is because we had a computational prediction. We tested in cell lines in a dish. We tested in a mouse model. And about 15 to 20 months after we got the computer prediction, we got ethics board approval, and we've started a clinical trial at Stanford on this drug, a clinical trial. And we all know it takes a long time to develop new drugs from scratch. 10 years, it costs $1 billion, $4 billion. If there's more use out there for these drugs, and computers and big data can help us get there, then this is how we're going to do this. And this is what we should be doing. And this actually led to a spin-out company called Numeti, which actually is really in the press today because the drug is actually now licensed to Numeti. And Numeti actually even showed that you can go beyond just being a platform play, but actually taking this platform, finding new drugs, and actually even licensing these drugs to pharmaceutical companies, which, by the way, are having a hard time actually picking up or even developing their own internal drug candidates here. Now, that's one kind of big data, those kinds of gene measurements. The genome is another big one, your DNA in all your cells. I think speakers already talked about it. I'm just showing a picture of the Whitehead Institute across the river here, which is one of the creators of the Genome Project, or at least created most of the data, much of the data. And many of you know that this used to take an enormous institution. The first genome cost about $3 billion, 13 years, and now they've got it down to a USB stick. Right? This is the USB stick from Oxford Nanopore. We'll see if this technology actually gets shipped at some point. But the point here is you can put now, in theory, your DNA onto a stick that's plugged into your USB port. And what took 13 years and $3 billion is now down to a couple $1,000s, eventually a couple $100s. And what we realized is we can start to interpret that data, but the interpretation was the hard part. So we started to read the papers, another source of data. As we open access to publications, we're going to be able to get the scientific findings. Here you're seeing 1,000 publications. This is the front page of 1,000 papers here on this grid here. We start to read 10,000, 15,000 papers, because what we realize is the hard part is analyzing the DNA. It's not going to be about getting the DNA sequence anymore. And so this is how we interpret the DNA. We come up with graphics like this. So one of the patients that came to our clinic, Steve Quake, who happens to be a faculty member, this is the kind of advice we can give to a patient now with their DNA. The black arrows indicate what the risk was for a particular disease before we knew the DNA. So for example, obesity, coronary disease, Alzheimer's disease. And the lines are either greater or less. Red means increased risk, blue means decreased risk, of now what the new risk is for these diseases given the DNA. And this is interesting because now for a 40-year-old or maybe an 18-year-old someday, perhaps we can start to tell whether a patient might have a problem with cancer, neurological disease, coronary disease, and perhaps try different treatment strategy, prevention strategies around it. That could be interesting someday. I get excited about kids like this. Now, if you haven't heard the story about Nicholas Volker, I strongly recommend you go look this up. This is a Pulitzer Prize-winning story, the first child saved by DNA sequencing. To make a long story short, 5-year-old kid who essentially lived in the hospital, had hundreds of surgeries because something was going wrong with his intestines, got a DNA sequence, parents got DNA sequence, they figured out a mutation. That led them to consider and actually deploy a bone marrow transplantation, and now this kid eats a normal diet and actually goes to school. And there's no way they would have figured this out if they didn't sequence this kid's genome. So in some ways, this is the first kid saved with a genome sequence. It's not just about the molecules. I think a couple people might have talked about Fitbits. I have mine as my prop here. I got my Fitbit. And so it's whether Withings or Fitbit. A lot of big data is going to come from small packages. Just because it's small doesn't mean it's generating big data. And in terms of behavioral change, whether it's organizational or individual, I think this little package is going to deliver a lot. Why do I say that? On the bottom here is my own BMI graph, okay? And you can see exactly when I started wearing a Fitbit and keeping track of what I'm eating. I was in the red BMI, and now I'm in the yellow, and I have a little bit more to go before I get into the green. Yes, I'm putting it out there. That's my BMI. But there's no way I would have been losing it if I didn't start to do this. I used to kind of laugh a little bit at the quantified self folks. And now I realize I am one of them. And actually made a hell of a difference here for me, and I think for my life and my health. It's also going to be about the cost data. You had a lot of people talking about costs and how the reporters are going to get to what we're charging for hospital care before the patients and administrators do, and come up with embarrassing stories sometimes in the newspaper where hospitals across the street charge 4 times more than each other. And it's going to be, in my view, the next big thing that I'm going to be working on is clinical trials data. Half of the clinical trials we run and pay for fail. They never even make it to a publication, and then they never make it into open access data. So we have this effort with the National Institute of Allergy and Infectious Disease, NIAID, and we've run this repository called Import, where we deliver clinical trials data, raw, individual level. It's de-identified, but maybe you can figure out which patients are benefiting a drug before the investigators actually did, and figure out patient subsetting and personalized medicine. And it goes on and on and on, whether it's EHR data, cancer genomes, ENCODE data, and all the rest. So what is big data to close this out here? What is big data in biomedicine to me? Is it about algorithms, programmers, databases, I don't know, high-performance computers? You've heard a lot of these things. Is it about mobile health? I actually don't think it's about any of these things. I think big data in biomedicine is about predicting the disease before it strikes, explaining the rare disease that defies experts, finding drugs for diseases lacking attention, making sure we do the right, safe things for patients, and big data is an amazing platform for biomedical innovation and even entrepreneurship. But I can sum all that up with one word. Big data in biomedicine is all about hope. It's hope for patients, it's hope for families, it's hope for providers, it's even hope for entrepreneurs someday. So if I had to sum this whole meeting up and what I think is a brilliant future in this field of big data and biomedicine, it's all about the hope that we're bringing to patients. I had to put in a plug for my own meeting. Colin spoke at my meeting. I'm going to put in a plug for big data for biomedicine next May. If any of you are on the academic side, think about coming to our meeting. I couldn't have done any of this with a long list of collaborators, a lot of folks helping with this mission. And I have to thank all the funding agencies, especially NIH and Lucille Packard Foundation for Children's Health. And wherever I go, I always thank my daughter, who lets me out of the house, and my wife, a molecular biologist, biochemist, who now starts companies with me, reads every major paper and grant that comes out of my lab, and lets me go all over the place like this to give talks. Thank you very much.